Hello everyone. Today we are going to be discussing a set of highly heritable behaviors referred to as innate behaviors. So let's do a bit of an introduction to this concept. You may have heard the term instinct before, and you probably associate it with behaviors that you're just born knowing how to do. Well, our formal term for those sorts of behaviors are innate behaviors. They're behaviors that animals are just born with, right? They're highly heritable and they're encoded for by genes. Here's a classic example. Nico Tinbergen, he was a Dutch-born British researcher who was one of the founding fathers of ethology. He actually shared the Nobel Prize in Physiology and Medicine in 1973 with two of the other founding fathers of ethology, Konrad Lorenz and Karl von Frisch. Lorenz we'll talk about later in this lecture. Von Frisch we'll talk about further on in the semester when we discuss communication. Now, Tinbergen studied a variety of different animals, but one of his most famous focal species were the herring gulls. Okay? We've got relatives of herring gulls here on the west coast as well. You've probably seen birds that look like this flying around at the beach. These gulls have chicks that look like this. They're very drab in color. Okay? But the adults, who are also drab, have this one brightly colored spot on their beaks. That spot is actually there for the benefit of the chicks. When chicks hatch from their eggs, one of the ways that they request food from their parents is by pecking at that colored spot on the adult's beak. That pecking is a stimulus for the adult female. The stimulus triggers the behavior of regurgitation. Regurgitation is when an adult has previously consumed a food source and then regurgitates it back up either from their stomach or from a storage pouch in their throat and feeds it to their offspring. And this is how chicks of most bird species are actually fed when they're too young to forage for themselves. So the way that chicks are fed in this species is they have to induce feeding from their mother by pecking at that brightly colored spot. Okay? For the chick, that spot is a stimulus. Seeing that brightly colored spot stimulates the behavior of pecking. For the adult, the pecking is the stimulus, and it stimulates regurgitation. Right? You get the idea? Now, the question is it just the red spot that triggers pecking behavior in chicks? Is it possibly a combination of the red spot and the shape of the bill? Could it be a combination of the red spot and the presence of an adult, a large bird nearby? Is it really just that one stimulus of red dot? Or is it a whole suite of stimuli that have to be present for pecking behavior to be initiated? Well, one of the things that Tinbergen examined was that very question. What does it take to get a juvenile herring gull to peck at a brightly colored spot? Does it have to be on the head of an actual adult bird? Can it be on the head of another bird? Can it be uncoupled completely from the head and still work? Well, here's what he found. If you present chicks with a head that has a red dot on the beak, so this was a little maquette, it was a little fake doll of a parent that had a red dot on the beak. The head features were all present. It looked like an adult gull. You got the result you would expect, which is 100% of chicks pecked at the red dot. Now this makes sense. It means that the models he was using were accurate enough that it convinced the chicks they were looking at an actual adult bird. Now, what's very interesting is if you show the chicks just a beak alone with a red dot, so instead of having the whole head present, you only have the beak and the rest of the head is gone, but you still include that red dot. Almost 100% of the chicks will peck at that as well. So the head, the eyeball, all the other features that make a gull look like a gull don't have to be present. just a beak with a red dot on it. Okay. Now check out the last result. Let's say you have a head with eyes, very realistic, complete beak, looks something like this over here, but it doesn't have a red dot. Look at how things change. Only about maybe 30% of chicks actually pecked at that head. Now that head, to us, looks a whole lot more like an adult gull than this disembodied floating beak does. But to the chicks, without that red dot, there's no stimulus. There's nothing to induce them to peck at this head if that red dot is absent. It's the red dot on the beak that matters. That's the context for the red dot that gulls recognize. There's another experiment that was done. It's not displayed on this chart, but it stuck an appropriately colored dot or spot on the end of a stick and then showed the baby gulls just this stick with, say, a red dot on the end. The chicks would peck at that as well, and at rates higher than they would peck at just the head with no red dot. Here's what's happening in that chick's little brain. Okay? The behavior of pecking is not voluntary. These chicks are not choosing to peck or not to peck. The neurons in their brain are reacting directly to the stimuli of seeing that red dot. This is a hardwired behavior, meaning it's encoded directly into the neuron's function. So the function of this particular cell includes responding to this stimulus. It's not learned, it's not observed, it's something they're born with. It's often highly stereotyped as well, meaning there's no variation in how the behavior is done. That pecking looks the same every time the gulls do it. There's no improvisation, there's no elaboration. That behavior happens the same way each time. And oftentimes, that behavior is uninterruptible, meaning once the behavior has begun, it cannot be stopped. We'll talk about what that looks like in a couple of slides. And finally, because this is a behavior encoded for by genes, it's highly subject to natural selection. Remember, natural selection operates on changing gene frequencies, just like all evolution does. If these behaviors are encoded by genes, it means natural selection can absolutely act on them. There are two major types of innate behaviors that we're going to discuss in this lecture. The first topic we'll discuss are reflexes, and the second are a really interesting set of behaviors called fixed action patterns. But we'll start by taking a look at reflexes. Reflexes are something that many of us are intimately familiar with. We have them, just like most animals do. They're innate behaviors, but they are involuntary. Okay? They're a response to usually a physical stimulus or a sensory stimulus, something that we see, something that we feel, and they're mediated by something called a reflex arc. Okay? The reflex arc is actually the pathway taken by information from neurons that are picking up on information from the environment, what we call sensory neurons. So these are neurons responsible for things like collecting visual information or sight, touch information, okay, physical. Those sensory neurons pick up that information. They send it to the spinal cord, which remember is part of your central nervous system. And the spinal cord then, instead of sending the information to the brain, sends a command to motor neurons directly, telling them how to respond. So this arc, this reflex arc, completely bypasses the brain, means it cuts it out of the circuit. Instead, the decision, so to speak, on how to respond to the stimulus is made by the spinal cord. But remember, the spinal cord doesn't think. It's not processing information. Instead, you can think of this like a series of dominoes. When that sensory neuron is triggered, that's the first domino that falls in the sequence. It hits the second domino, which is the message arriving at the spinal cord. That then triggers our third domino to fall, which are the motor neurons responding to that stimulus because they're sent commands directly from the spinal cord. Okay? This makes the reflex occur more quickly. That's the advantage of cutting the brain out of the loop. These reflexes are not perfectly instantaneous, but they almost are. They might as well be. This is the fastest way to react to a change in your environment. Okay? They've got really, really high survival value, meaning they boost your chances of survival immensely. Why? Reflexes happen in response to stimuli that signal things that might be harmful to you. Okay? So, for example, blinking. The blink reflex is a classic one. When your eye perceives something is heading for it, it automatically closes the eyelid to protect itself. The sensory neurons in the retina that see an object approaching then send that information to the spinal cord, which immediately sends back a command to close the eyelid. It's almost instantaneous, and it protects your eyes from damage. That's got huge survival repercussions. The startle reflex is another one that has strong consequences for survival. I'm going to show you a video here, down at the bottom. What you're going to see here is a mother panda's response to her baby sneezing. So clearly that's a pretty dramatic response on the mother's part. What you just saw is a classic example of the startle reflex. So the baby sneeze caught the mother completely off guard, and her immediate reaction was to jerk backwards, away from the baby. It's a whole body response. If you've ever gone through a haunted house attraction, you've had the same response when someone has jumped out at you to try to scare you. Your whole body jerks away from the stimulus. This is called the startle reflex, and it is highly, highly adaptive because it pulls you away from a threatening stimulus, which, if you were actually about to be injured, might minimize the damage to you as you pull away from whatever it is that might be lunging towards you or might be threatening you. Okay, so high survival value. Now, there are some other reflexes that might be a little bit less apparent in their value to your survival. For example, the patellar reflex is the reflex that occurs when your doctor or your coach or your physical trainer uses what's called a patellar hammer, this tool right here, to knock on your knee. Most people who have had a doctor's appointment, in the US at least, have had this reflex test done. What's happening is this hammer is hitting a cluster of what are called muscle spindles, and they're clustered right above your kneecap. Right below your kneecap. And the formal name for your kneecap is your patella. This little cluster of muscle spindles are receptors. They're sensory neurons. When they detect this impact, they send that message out to the spinal cord. Okay? The spinal cord then sends an immediate message back to the muscles of the leg, telling them to kick the foot forward. Okay? So you have the impact with the kneecap. That sensory information gets sent to the spinal cord. Spinal cord sends back a message to the muscles of the leg, telling them to kick. Now, you're kicking because when this test is done, you're normally sitting down with your legs dangling. But in normal life, this nerve cluster would be triggered by, say, jumping off of something and landing, walking when your foot impacts the ground, or if you are walking on unstable ground and sort of having to hop from foot to foot. Okay? Each of those actions creates an impact on the bottom of your foot that travels up to this nerve cluster and stimulates it. What happens then is the quadricep muscle meaning your upper leg muscle, also called your hamstring, overreacts. Okay? This helps you to maintain your balance. It puts sort of a bit of extra strain on your lower leg, which actually helps keep you upright so you don't just fall forward when you land after jumping off of something. This strengthens the muscles, tenses the muscles in your lower legs, and it helps keep you standing upright. Same thing when you're walking. Each time you put your foot down, you have the potential to just allow your knee to continue bending and to fall, to collapse onto that foot. However, this patellar reflex ensures that the muscles in your leg tense up when you put your foot down and allow you to keep that leg straight and to keep you standing. This also has pretty high survival value. You don't want to be collapsing every time you jump off of something and land on the ground, or if you're trying to maintain your balance and stay upright while walking or moving over uneven territory or uneven land. Okay? So these reflexes, they're innate, they're involuntary, they're a response to a stimulus, and they are controlled by this reflex arc. That's a connection between your sensory neurons, your spinal cord, and then your motor neurons. The next type of innate behavior we need to talk about are very different from reflexes. They're called fixed action patterns. So fixed action patterns were first formally defined by Konrad Lorenz, who's an Austrian researcher and one of the three men who shared that 1973 Nobel Prize I mentioned earlier. Fixed action patterns are behaviors that are highly stereotyped responses triggered by a well-defined, simple stimulus called a sign stimulus. Okay? The sign stimuli are very basic, okay? Very simple. I'll give you an example on the next slide. Now, fixed action patterns themselves, the behaviors themselves, are very stereotyped. They happen the same exact way every time. They also tend to be surprisingly complex behaviors. They're species specific, meaning they're unique to individual types of animals. And once the behavior has been started, once it's begun, it's uninterruptible, which means the behavior itself has to play out to its conclusion before the animal can do something else. The other thing about fixed action patterns is they're completely unlearned. Animals are born knowing how to do these things. Now, once the pattern is activated, the response is performed in its entirety. Right? So you can think of fixed action patterns as following this basic formula that you see in the box here. The sign stimulus is what starts it out. When sensory neurons pick up on that specific stimulus and that information reaches the brain, a specific kind of neural network called an innate releasing mechanism triggers the behavioral pattern itself. Okay, so this is a specific cluster of neurons in the brain. Let me give you an example of a fixed action pattern. Gray-lag geese were studied by Nico Tinbergen and Konrad Lorenz. They did work together on some projects. Gray-lag geese incubate their eggs until they hatch, like most birds do. But the trick is, the gray-lag goose has a fixed action pattern that is triggered by the sign stimulus of an egg being removed from the nest. So when that egg is removed from the nest, if it rolls away, the females see it and it triggers the fixed action pattern of retrieval. The goose will extend its neck. If you look at the upper right-hand diagram, the goose will extend its neck outwards, tilt its head downwards, place its beak behind the egg, and then drag the egg back into the nest. Right? So this is seemingly a simple behavior, but it's actually a fairly complex series of motions. The goose has to extend its neck outwards, angle its head down, tuck its beak under the egg, and then roll the egg back underneath it. That actually winds up being a fairly complex set of movements. Now what's interesting is this fixed action pattern will occur with any egg-shaped objects, regardless of, say, size. Let's look at this second diagram. This is an enormous egg. Obviously, this egg is a lot different from all the little ones that are here already in the nest. Doesn't matter. If this egg is placed outside the nest, the female goose will respond to it as though it were one of her own. She'll extend her neck, drop her head, tuck her beak under the egg, and roll it back underneath her. Okay? It's the egg shape that's the sign stimulus, not its size. Now, once this retrieval behavior has begun, it's going to continue uninterrupted, even if the egg is removed. So what you see in this bottom picture is, I believe this is Lorenz, grabbing the egg and removing it from underneath the goose's neck. So even though the egg is removed, what happens next is the goose's head continues to pull backwards, as though the egg were still tucked under its beak. Even though the goose is well aware that the egg is gone, it can't interrupt the fixed action pattern. The behavior has to be completed. Okay, that's something unique to these FAPs. And let me give you another example. This research was done by Tinbergen on his own, and it was done on a type of fish called the three-spined stickleback. Male three-spined sticklebacks are highly territorial, especially during mating season. Now, what's interesting about this kind of fish is the males, like you can see up here in the top picture, have this red belly that shows up during mating season. It's not there during the rest of the season, just when it's time to mate. And this is a visual signal to the males that it's time to start competing for mates and establishing a strong territory. Males identify each other using this red coloration, and they'll attack one another and duke it out for good territory or for the attention of females. The trick is that males will attack just about anything with red coloration as long as it's in the right spot on the object they're looking at. The sign stimulus in this case is a red belly. So the bottom of the object that they see has to be red, top has to be some other color. And it doesn't matter what the other characteristics are, whether the object is a funny shape, whether it has fins or no fins. If you look at the diagram on the right here, what you see is that the male sticklebacks will respond to any of the shapes you see on the bottom two rows. Doesn't matter what shape the object is, doesn't matter how large it is, so long as it has a red belly, male sticklebacks would respond to it aggressively. Now look at the diagram on the top. That's a pretty good representation of a stickleback. Models were made of all 5 of the objects you see here. So models were made of these strange looking shapes that just have a red base to them. And then a model was made of this very realistic stickleback that has no red on it. What they found is all of the models below that line elicited a response, strong response, from male sticklebacks. They were all attacked. But males did not attack the model on top, even though physically it has much more in common than an actual stickleback. But it doesn't have the red coloration, and that's what matters. Now, I've mentioned a couple of times that these fixed action patterns are uninterruptible once they've begun. Now, this could obviously be problematic if, say, your brain detects another stimulus that might indicate you should be doing something different than the fixed action pattern you're engaged in. The brain is able to organize behaviors based on priority, and that's what we're going to talk about next. So, what stimuli are organisms going to respond to, and in what order? How does the brain determine prioritization? Let's talk through an example first. Give you a little sense of how this works. Bats hunt using sound waves, which is what the lines in this diagram on the upper right are showing you. They use what we call sonar, so pulses of sound represented by these lines moving outward away from the bat and towards that moth. Sound waves then reflect, literally they bounce off of the moth and back towards the bat. So the bat sends out sound waves, which then reflect off the moth and come back, giving the bat information about distance, direction, and size of that moth. Also, what it's currently doing. This type of sensing information using sound is called echolocation. And it uses ultrasonic pulses, so super high frequency sounds that the bats produce themselves. Now, in this example, we are going to talk about this interaction, but from the moth's perspective, right? So you're a moth, you're flying along, and suddenly you get hit with this wave of sound. You're able to detect it using your antenna, these guys right here. Moths have a fixed action pattern that they've evolved to evade bats and try to survive these interactions. When the moth detects these sound waves coming at it, that sign stimulus gets sent to the brain, the information about it gets sent to the brain. The brain then sends out a motor command to the wings that cause the wings to tuck back against the body. Okay. This turns the moth essentially into an oblong bullet shape, okay, with its wings tucked tight against its body, and it drops like a rock, falls right out of the sky. This is the quickest way to get out of the bat's field of view. So when the moth gets hit with that sound wave, tucks its wings, it drops straight down, and the bats lose track of it. So your fixed action pattern here is wing tuck, and drop. The moths will eventually open their wings again and start to fly. They do this before they hit the ground, but they'll drop a significant distance first. Now there's another fixed action pattern that moths exhibit, and it's very, very different from the one that helps them evade predators, especially bats. Male moths are hypersensitive to female moth pheromones, which are chemicals that signal female readiness to mate. The pheromones, in this case, are the sign stimulus. And the fixed action pattern is to fly towards the source of the pheromones. So that approach flight, where the male moth is tracking where the pheromones are, making sure that he's flying in the direction of their origin, that's the fixed action pattern behavior itself. Now these are both fixed action patterns. Once they've begun, they cannot be interrupted. Here's the trick. There's an exception to that rule. The nervous system is able to prioritize one of these fixed action patterns over the other. If the male moth is currently flying towards a female, and suddenly that male moth gets hit by ultrasonic waves, it will abort the flight behavior that's taking it towards the female, and instead it will tuck its wings and drop. So this seems to violate our rules. We said fixed action patterns couldn't be interrupted. Well, it turns out they can be overridden if a more important fixed action pattern needs to take place. Okay? This is called behavioral prioritization. Okay? The next question is, how does the brain do this? How is this achieved? Well, there's a variety of mechanisms that can allow your nervous system to prioritize certain innate behaviors over others. One example is neural control through what are called command centers. Okay? Command centers are clusters of nerves that control individual behavioral responses. For example, those IRMs, those innate releasing mechanisms, we talked about controlling fixed action patterns. They're an example of command centers. Now, what's interesting is that these command centers can mutually inhibit one another, meaning they can turn each other off if necessary, if the activity of both command centers at the same time is going to produce contradictory responses. Okay. So, for example, fleeing a predator and approaching a mate can't happen at the same time. Right? Moths tend to fly in a straight line towards the female. That makes them really, really easy to pick off by a predatory bat. Those two fixed action patterns are mutually exclusive. They contradict one another. Okay. Command centers are able to turn each other off in order to prevent those contradictory behaviors from happening simultaneously. Another example of a way that your nervous system can organize and prioritize behaviors are through what are called clock mechanisms. Clock mechanisms are another neural action. They can change the inhibiting of command centers based on environmental information. Okay? So, for example, it only makes sense to be territorial for three-spined sticklebacks during mating season. Doesn't make sense to be territorial at other times. So, clock mechanisms inside the brain can keep track of the seasons, the time of year, and ensure that fixed action patterns geared towards aggression and territoriality are less likely to occur when it's not mating season. And these clock mechanisms will prioritize other fixed action patterns over the aggression and territorial ones. And finally, our old friends, hormones. Remember that environmental conditions influence the release of hormones. And remember, hormones can cause physiological and behavioral changes. Really what they do is increase the odds of a certain behavioral response to a stimulus. Right? And that alone can change the prioritization of that stimulus. There's one more way that your nervous system can help to prioritize behaviors, and it's actually by prioritizing the stimuli that would elicit the behaviors. This is called stimulus filtering, and it involves limiting the perception, the ability to perceive certain stimuli, depending on different types of cues. Essentially, what that's saying is it tunes out noise. Animals are only really responding to a small subset of the information that they're taking in. We don't respond to every single thing we see. Instead, we filter through all of the images that we see when our eyes are open and focus on just the images that are relevant to us. We don't see all the decorations in a room. Instead, we see the ones that are interesting to us or that stand out. We don't hear every single voice and every single word in a party. Instead, we hear the conversation that we want to be listening to. So here's a couple of other examples. Some species of insect make their sounds in a specific auditory range. So say that cricket in the upper right corner is producing sounds that are very, very high frequency. Other crickets of that same species are going to have receptors that are more sensitive to that high frequency range and less sensitive to lower frequency sounds. That's stimulus filtering. The crickets can't even detect lower frequency sounds that aren't relevant to them. Star-nosed moles are another good example. They have a tactile mode of detecting information from their environment. They have those protrusions from their nose that give them a whole lot of sensitivity to the dirt and environment right in front of their faces. Okay? They live underground, they're subterranean, very, very little light. Look at how small their eyes are. They're not using vision to detect what's going on in their environment. They're normally using touch. Significantly more of the brain is dedicated to tactile stimulus processing, touch stimulus processing, than visual processing. And it's because of that stimulus filtering. The brain doesn't spend much time detecting or processing visual information because it's not as useful. Instead, the brain dedicates its time and energy to the stimuli that are most informative, which are tactile stimuli. All right, that's it for our innate behavior lecture. I'll see you next time.